Hey Taylor, miss you, hope you're doing well. So I'm gonna run through this because I gotta go, okay? But hopefully this helps. All right, so let's talk a little bit about this. Pedigrees. All right, so the cool thing about pedigrees is, and why they're important is especially for dogs. Like you kind of want to know, is your dog a purebred or not, and what kind of genes does it have, what kind of genes does the ancestors have, etc. So if you're a dog breeder and you really want to know if the dogs that you have, or let's say even if you're a farmer, you want to know what the plants you have, what kind of genes do they have, all right? You want to use pedigrees. So essentially what you do here is this. You just map out the traits of all the offspring and of all the ancestors of whatever organism it is that you're breeding. And so if we look at this pedigree here, basically the basic idea is this. Look at your key here, right? We have the boxes here are males, the circles are female, okay? But we really want to look at this. Affected, all right? And of course deceased means a line through, but you have affected and then you have the non-affected ones, which are the clear ones. So let's take a look at this, all right? So if we look at these two parents up here, the grandparents I should say, all right, both of them, all right, are unaffected, okay? So if we look here at their offspring, you would expect a couple things, right? Number one is if both of these were considered to be recessive, all right, then you would expect that all their offspring would also be recessive. All right, and all the offspring have to be recessive because both parents were, and that means the only genes were recessive genes, so they can only pass recessive genes on to their offspring. So offspring would also be clear. But that's not what we see here. What we see here is we also have these two, okay, affected males. So when we come back over here to this, we say, okay, so what that must mean is these both are not recessive, these both must be dominant traits, okay? They both must be dominant because we have here dominant traits and recessive traits, and this one has to be dominant, otherwise none of this makes sense. I feel like I'm stumbling over my words, but that's the general idea. Now, if you have two dominants, right, and they have dominant offspring, but they also have recessive offspring, that means both of them must have also had a recessive trait. They both must have been heterozygous, okay? So if you're looking right here, that would explain why one allele from mom, one allele from dad, or one allele from dad, one allele from mom, and that would come together to make this offspring right here. Same thing right here. One allele from dad, one allele from mom, makes both of them recessive. This is one of the ways, again, we can tell, all right, what the genes are of a particular group or species or breed that you're trying to work with, okay? And it's just a matter of using logic, what must make sense in your pedigree. But anyways, continuing through here, you can make some other predictions about the rest of them here. If you'll notice, assuming that this, again, is recessive, if we're looking at these offspring, we have some recessive, some that are dominant. So that must mean this guy over here, if it was homozygous dominant, then every single offspring would get a dominant letter, and this would not work. So therefore, it has to be heterozygous, okay? To make sure that you end up with, again, one allele from dad, one allele from mom, pairs up to make the offspring, okay? So it's a matter of using logic. So this should be another little closed example. If you're looking right here, we have two clears making one clear and one filled in offspring. So these two must have a secret or hidden gene. That's that recessive gene. They must have one secret or hidden gene because that makes its way here. Okay, but since they're both dominant in expression, they both must have at least one dominant or expressive gene there. And it comes down to this idea of this hidden second gene. The recessive one's always covered up or masked by the dominant one. Okay, and that's what makes this. So again, looking through here, I mean, we can't tell for sure what's going on here. We have to assume this is probably dominant of some kind because that's why this one's also dominant. And over here, again, this is likely a situation where both are heterozygous again because you end up with a mixed group of offspring, okay? So we see two recessive A's for both of these and then some kind of dominant in that one right there. So that's the whole idea of pedigrees. It's just kind of showing what the parents were like. There are other things they can do to represent it, right? Sometimes they can even include a cross box like this, which basically means you're a carrier. So over here, we couldn't tell if they were a hybrid or not. It didn't really show that. But you can represent it, though, with this little box. It would mean that one trait and the other. They would be heterozygous. That's what that would mean, essentially, okay, for whatever that trait is it's carrying. And just to kind of go over the finer details now that I realize I'm really talking about it, this right here, the ones at the very top are the parents, and they're connected by a line, and then it goes to their, in this case, 3 offspring.
Please pardon interruption. Can I get a custodian to the office, please? Can I get a custodian to the office, please? Thank you.
So just one more roundup here is kind of explaining it, okay? We have affected, we have unaffected, or whatever, however it is they're choosing to label it. But the point is this: if we're looking, and I think those are actually labeled backwards, but whatever. If we're looking right here, both parents are heterozygous. How do I know they're heterozygous? Because if I look at their offspring, the whole idea of pedigree is to look at the offspring. And I see the offspring here, right? Dominant, 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 okay? But this guy over here is recessive. And those 2 recessive alleles, one comes from mom, one comes from dad. Therefore, both parents must have had one recessive, okay? And that's the point here, is again, predicting what the parents are like. That's the point of the pedigree, okay? I have no idea if that helped. I've never taught this before, so if it didn't, you can always send me a message, okay? All right, so another one of the standards here is talking about the relative advantages and disadvantages of sexual and asexual reproduction. Okay, so talking about sexual versus asexual reproduction. So the basic question I have for this, and that you should be able to answer, is why are people different? Apologize for the interruptions.
Can I get Miss Hodges or Mr. Smith to the front office, please? Miss Hodges or Mr. Smith? Thank you.
So the question is, when we look at people, why are they all so different? Why do we have all these different types of noses, eyes, eyebrows, chins, hair color, eye color, face shape? Like, why are all these things different from all these people? And the answer is because of this. Oh, that's not going to write. Okay. The answer is because of evolution. Okay? When we talk about a group or a species, okay, what you want is every single species, for the most part, that engages in sexual reproduction. And again, that's what humans do. We do sexual reproduction. The whole idea of everyone being different is so that when we reproduce with someone, our offspring will also be different. It's bringing in this diversity. Because diversity can help a species evolve. The crazier the traits that you have, the more likely you might be to survive, right? If you're born 7 feet tall, and all of a sudden, you can only eat fruit from like super high trees, well, that 7-foot person's going to have an advantage. Like, being different and having unique things is the point of evolution, and genetics proves it, because everything in genetics is designed to create more diversity. Okay, that's what you're learning about right now in genetics, is how is this diversity created and why. So let's talk about reproductive strategies. You have 2 types, asexual and sexual. So in asexual reproduction, there is a single individual who is the parent, and that one person has but one set of DNA. So therefore, every single offspring will have the exact same DNA. That means there are no differences between the offspring and the parent, which means there's very little chance for evolution and adaptation. Okay? What makes animals special, especially, well, not just animals, but most organisms special, is the ability to do sexual reproduction. And this has a lot of benefits. Okay? In this situation, 2 parents give rise to offspring, and each one passes on half of its genes to the offspring. Okay? And by sharing their DNA and genes, their children are a mixture, right? So my child Maverick, for example, is half me, half of my wife, right? And so in that way, he's made better, in his case, right, than me. My wife, good eyesight, smart, beautiful, you name it. My son, also smart, beautiful, good eyesight. Me, all of the opposites of those things. Okay, so that's the point of sexual reproduction, is to create an offspring that has certain advantages with all of these differences in there. Okay, now, as we go on here, I think we talked about the rest of this, okay? All right, so if we're looking over this, advantages and disadvantages specifically, sexual reproduction versus asexual reproduction, again, the main thing is in sexual reproduction, you're going to end up with a high genetic variability. In other words, the more differences we have, the better. It speeds up evolution. Differences are good. We often talk about being different. Sometimes people view as bad or whatever. Well, you shouldn't. Being different is good, and it helps us evolve as a species. The problem is, right, the whole idea of reproduction itself, finding a mate, keeping a mate, reproduction itself, all of it costs energy. It's very time consuming. Okay? So, yeah, one sex is usually a little bit weaker than the other, which can cause issues. So all of these things can affect and be a disadvantage for sexual reproduction. Over here with asexual reproduction, the nice thing is it saves energy. You don't need to find a mate, right? Courtship is a non-issue. You just reproduce when you're ready. The problem is the genetic variability. There's very little actual fuel for evolution here. Okay? Yeah, retards evolution, slows it down. So how do we get those genetic differences in our offspring? So everything we learn about in genetics is how those genetic differences are made. How do we get people to look different? And it's through this. Sexual reproduction is a sperm and an egg. Okay? And the way they are created and the way they come together all mean that you could have differences. And that's the driving law of evolution. So this is the big standard here. Use Mendel's laws to ask questions and define problems that explain the role of meiosis in reproductive variability. Again, meiosis produces variability. That's the engine for evolution, that reproductive variability. So coming over here, this is all the basic stuff about genes that you should already know. I borrowed these slides from Miss Griffin. So if there's anything on here that you see, feel free to read through. I would encourage you to do so if you need help with the basics, right? The idea of the genotype versus phenotype, just the genes in the DNA versus the phenotype, the physical expression, okay? So make sure you're aware of what those are. These terms, homozygous, heterozygous, they're key, but you learned a lot of these in 7th grade. But if you want me to go over them in more detail, you just got to let me know, okay? But homo, right? You talk about homo sapien, you talk about homosexual. Homo just means the same, right? Someone is homosexual like someone of the same sex. You have hetero, so hetero means in this case different, right? Someone who's heterosexual, they like someone of a different sex. And when you look here at the genotype, that bears out. Homo means the same. Both of these are uppercase letters. Homo here means the same. Both of these are lowercase letters. Hetero means different. One uppercase, one lowercase. And just to remind you, the dominant one always wins. So in both of these situations, you have a dominant letter, therefore a dominant physical trait. That's the phenotype. The gene, right? One allele from mom, one allele from dad, and the phenotype, right? The physical expression on the child. Okay, genes are inherited two following ways. Gregor Mendel, the father of genetics here, talking about the law of segregation and the law of independent assortment. So these are pretty big, okay? But they're not hard to understand. I promise you they're not. And you kind of already know or can figure these out based off the words. So use your social studies understanding if the science is leaving you behind. This word segregation, you know what segregation is. It's what white people did to black people back after slavery, right? They instituted segregation, right? They took this group over here and said, you can live here. They took this group over here and said, you can live here. And that's what segregation was. You can use this, you can't use that kind of thing. So if we're looking at genes, genes can be segregated as well, all right? So if we're taking a look at this, it says gene pairs separate when gametes are formed, so each gamete only has one allele of each pair. Like if you're looking at your chromosomes here, these X's that you can see, okay? If you just split them up, okay, and put them all in their own unique packaging or section, you are segregating them by splitting them up. Does that make sense? This idea of segregation. You're only going to get one chunk here, and this one chunk will go to your offspring. Your offspring could get this chunk, it could get this chunk, it could get this one, it could get this one. It doesn't matter. It's getting one of these chunks from the parent, okay? So this is the parent. It's got the basic, let's say this one is heterozygous like this, okay? So this is a big A, this is a big A, this is a little A, this is a little A. This is what the parent looks like, okay? And when it looks at its offspring, these are the 4 possible segregated chromosomes or chromatids that could go to the offspring. All right? Segregation, dividing it up into sections. The law of independent assortment does the following. Again, just looking at the words, independent assortment. An assortment of goods. You look at a bag of trail mix. That's an assortment of goods. They're different things. Okay? Put together. Different things being put together. Okay? So an independent assortment, all that means is these things are being put together, okay, in different ways. Different pairs of genes separate independently of each other when gametes are formed. They are independently sorting themselves into their unique place. If we look at it like this, independent assortment says this. Once you segregate and separate these things, then you can independently assort them. In other words, just stick different ones together. independently. Okay? Like, look at it like this. On this image, if I can get this, maybe it's a little bit bigger. Okay, so if we're looking right here, what I mean by independent assortment is essentially this. You get different flavors of each. Each little package has its own unique expression. Looking at these genes to start, right, they get segregated and put in their own sections, and then they get independently assorted. So one red, one green, one red, one green, one blue, one yellow. They're independently assorted into their own unique place and space. Okay? Oh, that's too much. Excuse me. Okay. Maybe I can get it to go a little bit bigger. A little bit bigger. Okay. Now, moving on over here. Using math models to predict and explain patterns of inheritance. So this is your Punnett Square stuff. So believe it or not, these Punnett Squares of yours are going to rely a lot on segregation and independent assortment. The way these different genes kind of pair up together. You'll see a lot of that in here. But yeah, we understand the colors of traits and how that's affected. I'm going to move on here. Punnett Squares you should be familiar with, all right, from 7th grade, and they're pretty straightforward. You have the parents' alleles on the top, right, and you have the offspring's alleles are in the inside boxes here, okay? The possible offspring are all in here. So move from the top down, right, top down here, and then from left to right, these all pair up until you get your offspring here. So this is something you could probably do really well in 7th grade, and you probably still can do it really well now, okay? But the point is this, when you're looking at this, you're talking about probabilities. There is a 3 in 4 chance of ending up with tall pea plants, but there's a 1 in 4 chance of ending up with a small one. So if you have 100 offspring, right, 75% of them, or 75 at least, or somewhere around there, are going to be tall, and 25 of them or so are going to be short. That's going to play out every single time. That's what these genes and laws are about. But the problem is, this is the easy bake oven stuff with just what we call a monohybrid cross. One set of genes to discuss, okay? What you want to be able to do now in high school is do what we call dihybrid crosses. And you can, of course, pause this anytime and look at some of these slides if you need help. The monohybrid cross, right? Just one gene, mono meaning one. A dihybrid cross means we're looking at 2 genes now, okay? And so in this case, we have YYRR and YYRR, all right? So let's look over here at this. I think this is probably a good place to start. So parent 1, homozygous dominant. Yes? Parent 2, homozygous recessive. And the question is, can we make a Punnett square that deals with 2 sets of genes? Okay? And the answer is quite yes. But how you set it up matters. And this is all about this idea of segregation and independent assortment. Okay? Let me explain what I mean. When we talk about segregation, we talk about separating these things, right, into their own unique categories. Okay? So we can separate them. We separate the Y's and the R's. Okay? Then there comes this idea of independent assortment. In other words, of these 4 potential offspring, these things can independently assort. In other words, these things, the individual letters can go in individual places. So this Y could go here, and this R could go here. Okay? This Y could go, this Y could go here, and it could partner up with this R. Okay? This Y could go along with this R, and this Y can go along with this R. It's either or. Okay, but there's 2 possible options for that one. However, this Y cannot go into the offspring with this Y. There will only be one Y, okay, and one R. So over here, I could also have this Y and this R pair up. Right? I could have this Y and this R pair up. They're all independently assorted. They're going into their own unique packages in unique ways. And that's what we're going to see here when we do this. Let me try this. Let me give you a better example here, maybe with different types of letters. So if we do, let's do A's and B's. Okay? So if I cross, we'll call this a dihybrid cross. And what I'll do is I'll cross 2 heterozygous parents. Okay? Both alleles are heterozygous. One is going to go on the top and one's going to go on the side. The question is, of course, what do I put where? Well, I have to remember this idea of independent assortment. All right? I have to make sure that I look at the possibilities of what the offspring could get. If I look at this parent, okay, and I look at what this parent could give its offspring, remember this is the offspring box right here, right? This parent can give one A and one B, one A and one B only. The question is which A and which B, and we don't really know. So what we do is we model it with these Punnett squares to make the possible outcomes. Just list out all the possible outcomes. So I'm looking at this adult here, okay? It could give the following. It could give a big A and it could give a big B. That's a possibility, right? So over here, I start with that big A, big B. It could also give me this big A. It could give the offspring a big A. It could also give the offspring a little b, right? It could be from either one of these. This A, big A, could pair up with either one of these. So that's two possibilities here, okay? Over here, now I've got this little a, all right? And that little a could partner up with the big B. That little a could also partner up with the little b. That's how you determine the top of your Punnett square in these dihybrid crosses, is by what are the possible assortments I could have, right? The different groupings I could have from this right here. And I'll do the same thing with this one, and I'll do it over here on the side. All right, so again, first of each. I think you might have done FOIL maybe in your math classes before. First, outer, inner, last. That's kind of what this is. First, right? You could do outer, big A, little b. Yeah. You could do inner, and you could do last. And that's how you get that. It's the same basic idea. Multiplying binomials or doing dihybrid crosses. It's all about two pairs of things and the possible combinations that they could make. Okay? But once you have this, then it's a matter of moving up to down. These all move up to down. All right? And so you can fill them in. Big A, big B, big A, big B, big A, big B, big A, big B, and on and on and on. Over here, same thing, up to down. And then you fill in these ones from left to right. Yeah? And then this would be a little b, maybe over a tab. And then we have the little a and the big B. And then we have the little a and the little b. Okay, and that's how you get, again, back to the original looking genes here with 4 unique units. It's because you looked at the possible outcomes for all offspring that are independently assorted. Okay? I won't get into test crossing because that's just too much. All right? But if we take a look right here, just to give you a basic question, which of the following genotypes would you not expect to find among the offspring of S, S, Y, Y, and this set cross? So if we cross these, okay, which of these would we not expect to find? Well, let's take a look. You don't even have to really do the Punnett square here, but you can, okay? You don't have to do Punnett square because if you're looking, this is homozygous recessive, okay? So if we're looking over here, this could never happen. You see there's 2 big Y's? So it can give one big Y from this guy, but there's no big Y over here to partner up. So this one could never happen, all right? But if we decide to do it regardless and set up our Punnett square, remember independent assortment takes one letter, right, from each of the 2 different types. So big S, big Y. Okay, ooh, sorry, I put it in the wrong place. Big S, big Y. I could put a big S and a big Y. I could do a big S and a little y. All right, I could do a little s and a big Y. And then I could do a little s and a little y. Again, putting all possible combinations. First, outer, inner, last. FOIL. Same basic thing. And then I do the same thing for this one. But all of these are little. So little s, little y, little s, little y, little s, little y, little s, little y. And then I just pair them up. And then little, 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 little. Okay? And I do that for all the rest of them and pair them up. And if you look and see, you will never find one like E. in that big list. Okay? And so that's how you do those big old giant dihybrid crosses. And the last thing I would say is this. You recognize those numbers right there? You should know that. That's from what's called a dihybrid cross. That pairing of numbers of possible offspring only happens when you have two heterozygous parents. Okay? If you look right here, this is not heterozygous. Okay? And so this one right here is not heterozygous. So this would have to be to give you that. So that's an easy way to simply look for it. 9331 is always with two hybrids. Okay? All right, I'm tired. I really hope this helped. I couldn't really find the teachers here. They have, you know, their notes and lectures. We don't really have too many resources yet, and I haven't taught this before. So I really hope that this helped. I don't know if it did because I've never taught it before, so I don't know what misconceptions to address just yet. But my best response to you is, if you have any issues with any of this, just send me an email saying, hey, could you tell me how this works or why this works, just so I can update this for you. Okay? Hope this helped. Have a good weekend. I'll see you later. Okay?